Hello, my name is João Felipe and this is the presentation of my thesis. In this presentation, I will first introduce the life cycle of experiments and present the necessity and challenge of provenance from scripts. Then I will discuss the studies we carried of the state of the practice on the usage of scripts and notebooks and the state of the art of provenance from scripts. Then I will present the approach we proposed for collecting, managing, and analyzing provenance from scripts and notebooks, and I will conclude the presentation. The life cycle of experiments is composed of three main phases: composition, execution, and analysis. During the composition phase, scientists formulate hypotheses and compose execution plans. In the execution phase, they execute the execution plans over the input data, which represents a context or population. And in the analysis phase, they analyze the experiments and visualize the results, seeking to elaborate conclusions. Depending on the results, they may repeat the cycle in a trial and error process, adjusting the execution plan and trying different input data. For composing the execution plan, there are three main strategies. Scientists can use system programming languages such as C and Java. They can use scripts such as Python and R, and they can use scientific workflow management systems. Each one of these have advantages and disadvantages. In this thesis, we focus on scripts. Scripts have the advantages of supporting gluing code, providing sophisticated data structures, supporting automatic memory management, being flexible, and so on. And they have the disadvantages of being slower than system programming language and lacking the provenance support that exists in scientific workflow management systems. But provenance is important for experiments because provenance helps to answer these two trial and error questions, these two questions that occur in the trial and error process of the life cycle of experiments, which are which part of the input data influenced a given result and how was the execution flow of each trial. Provenance refers to the documented history of an art object or the documentation of processes in a digital object's life cycle. Besides answering these two questions, provenance has also other usage in a scientific context. It can be used to share results, reproduce experiments, check the integrity and authenticity, understand experiments, and in a general context, it can be used for detecting system dependencies, intrusion, system change, and it can also be used for debugging and education. So it's important to have provenance, but it's challenging to collect provenance from scripts because scripts have control flows and cycles, so it's hard to uniquely identify constructs that occur inside loops. Also, scripts lack well-defined concepts for activities and data. Scripts can have functions as first-class citizens. So, for instance, here we have the variable first in line 5 acting as a data and in line 8 acting as a process, as a function call. There are also the challenge of deciding who should collect the provenance, should be the user or an automatic system, and at what granularity should you collect variables, function calls, files, and depending on the decision, you may have false positives and false negatives. And also scripts run outside controlled environment, so you cannot make any assumptions about the environment that the user running. For writing scripts, there are three main tools. Scientists can use text editors, IDEs, and interactive notebooks. Text editors are generically simple and lightweight, but they often lack development features that exist on IDEs. IDEs have specific features for some programming language that try to improve the quality of the development, but they lack exploratory and scientific features. So often scientists use interactive notebooks, which are tools that adopt the iterative programming paradigm that combines formatted text, executable codes, and execution results, and they use the interactive notebooks for exploratory research. However, notebooks have some characteristics that has been criticized for encouraging bad habits that may impair the reproducibility of the experiment, such as hidden states out of other cells, and the format does not encode library versions. Hidden states occur when the stored notebook results do not represent the notebook definition. So here we have an example. Let's say we start executing the first cell of the notebook, then we execute the second cell, incrementing value, then we execute the second cell again, incrementing value again, then we execute the third cell, and the result of value is 2. However, if we execute this notebook following the execution order again, the result of value will be 1 because we incremented it only once in the re-execution. Also, notebook supports executing the cells in a different order, so it's possible to start executing the second cell and then go back to the first cell and then execute the third cell, and the result of value here will be 5. However, if we execute in the order, the result of value will be 4, so it's also a problem for reproduction. Also, the notebooks have the imports in them in the code, but they do not specify the version of the import. So here we are importing matplotlib, but depending on the version of matplotlib that we have installed, it may cause a deprecation warning, an error, or even produce a different result without notifying the user that it's different. So we need to know what are the versions of the modules. We could use some external files such as setup.py, requirements.txt, and pip file, but the notebook does not encode the versions. And in addition to these challenges, there are the provenance challenges from scripts, because notebook run scripts, so they also have all the provenance challenges that exist in scripts. So the hypothesis of this work is that scripts can also be supported by an infrastructure for collecting, managing, and analyzing experiments, respecting constraints of usability, effectiveness, and efficiency. And we have 4 goals. The first goal, to understand how scientists use scripts and notebooks. The second goal is to identify approaches that have been proposed to support provenance from scripts. The third goal is to propose a tool that collects, manages, and analyzes provenance from scripts for supporting understanding and reproducibility. And the fourth goal is to propose tools that collect and analyze provenance from interactive notebooks for supporting quality and reproducibility. So let's start with our first study on the study of the practice of scripts. And the goal of our study is to understand why and how scientists use scripts in experiments. And for that, we ran a questionnaire with 120 scientists, and we analyzed Python scripts from data 1. So in the questionnaire, we had the goal of answering these 3 research questions. We will not discuss them in much detail here due to time constraints, but the discussions are available in the thesis. So the first research question: what are the scientists' preferred or more often used tools to run experiments? And the participants of the questionnaire answered that they use Python, Shell script, and R, which are all script language. And we also found that they do not use the scripts by themselves. They also combine scripts to system programming language and to scientific workflow management systems. The second research question, which tool is the favorite? We found that Python was the favorite tool selected by the scientists. And in the third research question, what are the reasons for the preference? We found that the scientists prefer scripts due to their flexibility, expertise, availability, and learnability. In the script analysis of the scripts from data 1, we collected 172 Python scripts and tried to answer 3 research questions. How do scientists structure scripts? And to answer this question, we extracted the abstract syntax tree of the Python scripts, and we observed that scientists use simple constructs in most cases, such as imports and loops and conditions and built-in data structures, and they do not use much other constructs such as function definitions, class definitions, comprehensions, exception handling. We also observed that scripts have 4 main regions in them. They have a header where the scientists describe the experiment and import the modules. They have a top region where they define parameters. They have a definitions region where they define functions and classes, and a bottom region with the main code of the experiment. In the second research question, how do scientists use modules and external tools? We categorized the modules and external tools that we observed in the scripts according to their domains and types. And we found that built-in modules appear in most scripts, but some external modules such as NumPy and Bio also appear in many scripts. Finally, to answer how do scientists process data in scripts, we observed many characteristics, and among them, we identified the 4 strategies for processing the data: during the input, during the output, in the middle, and interweaving input and output. So in this section, we observed that Python is the favorite tool among participants due to its built-in collections and modules and the flexibility with support for tool integrations. We also observed that scientists use simple constructs in general, but there are some scripts that use complex constructs such as functions as first-class objects, and we found almost no evidence of provenance in scripts. In the next study, the state of the practice on notebooks, we tried to answer 7 research questions. Here we will focus only on the last 3. So the research question N5, do users store notebooks with executed data? To answer that, we analyzed whether the notebooks have the execution data stored in them and which output formats do they use. In the research question N6, how are notebooks executed? We observed the execution count that appear next to the cells and counted the number of skips and out of other cells to observe these situations that cause hidden states and out of other cells. And to answer the research question N7, how reproducible are notebooks? we observed how many notebooks belong to repositories that do not have requirement files, and we ran a big reproducibility study where we tried to execute the notebooks and check how many of them produce the same results. For this study, we collected over 1 million notebooks from GitHub, and we applied selections over them for each research question. So, for instance, to answer the research question N7, the reproducibility study, we selected 750,000 Python notebooks with unambiguous execution order. That is, notebooks that do not repeat the execution count of the cells and also that have execution counts in them. We also select a subset of popular notebooks based on the number of stars and forks in their repositories, and we sampled notebooks for a qualitative analysis. So, answering the research question N5, do users store notebooks with executed data? We found that yes, 96% of the notebooks had at least one cell with an output, and 54% of the code cells had an output. This result is important for reproducibility because it indicates us that we can re-execute the notebooks and compare the results that are stored there with our results. In the research question N6, how are notebooks executed? We found that 76% of the notebooks have skips, which may indicate hidden states, and 36% of the notebooks have out-of-order cells. We also found that users pay less attention to the bottom of the notebook. The bottom of the notebook has less executed cells, it has more empty cells, and it has less markdown cells. So all these problems indicate that the reproducibility may be impaired. Finally, in the research question N7, how reproducible are notebooks? We first analyzed how many notebooks belong to repositories that declare dependencies, and we found that most notebooks do not belong to repositories that declare dependencies. And when we tried to execute the notebooks, we could only run about 25% of the notebooks. And by running the notebooks, I'm considering running all cells of the notebooks, because we could run one cell or another, but not all cells. And the results of 25% occurred in different environments following different execution orders and different dependencies. But this result is similar to the result that Kohlbeck et al. Found for software from our general computer science research. They also could only run 25% of the computer science research software, and they didn't check the results. They just tried to run the software. In our case, we also checked the results, and we found that about 5 to 9% of the notebooks produced the same results. And when we normalize the results, the reproducibility rate reached 11 to 15%. And the main reasons for failure are missing dependencies, the presence of hidden states and out-of-order cells, and data accessibility problems that occur when the users use absolute paths and when they do not include the data in the repositories. So in this study, we found evidence of good practices and bad practices in notebooks. As good practice, we found the usage of literate programming. We found that notebooks are stored with results. We found that notebooks use abstracts when they need complex control flows. And we found that notebooks use descriptive file names. As bad practices, we found the lack of dependency declaration and the presence of out-of-order cells, non-executed code cells, hidden states, and we found the lack of tests. The results of this study were published in MSR and received 2 awards, and in EMSE. Moving on to the study on the state-of-the-art of provenance from scripts, we ran a systematic mapping that started by a Google Scholar search of scripted provenance, and we found 90 papers. And from these 90 papers, we exhaustively ran forward and backward snowballing and visited over 1,000 unique references and selected 53 papers referring to 27 approach. We categorized these approaches according to their main goals, and this is the result. And we found these 5 main usage goals: caching, comprehension, framework, management, and reproducibility. And we analyzed this approach, understand the characteristics of them, and organized them in a taxonomy. And this taxonomy has 3 main branches: collection, management, and analysis, which we discuss in more details in the thesis. After publishing the taxonomy, new approaches have been proposed, so we updated the literature review by an exhaustive forward snowballing, and we visited 600 new references. We selected 33 new papers and 17 new approaches. So in this section, we identified 5 provenance applications, we proposed a taxonomy, and we observed that few approaches collect fine-grained provenance. None of them support generic complex data structures, but some support fine-grained collection of changes in data frames, and some collect data structures as a whole. We also observed that few approaches consider the provenance evolution. Some approaches use version control systems for that, but they do not define a version model for provenance. And few approaches support clustering and filtering. Most approaches just display the provenance as direct graphs as is. And the results of this study were published in a paper in ACM Surveys. Next, based on the previous results, we propose approach that collect, manage, and analyze provenance from scripts. Our approaches are based on Workflow Zero that was proposed in 2014 to collect provenance from Python scripts transparently. That is, it does not require changes on the scripts. In this thesis, we propose 2 new versions of NoWorkflow. NoWorkflow 1, that is a direct evolution that supports fine-grained provenance, graph comparison, and provenance evolution, and NoWorkflow 2, that is a revolution that is less intrusive, more precise, and have efficient algorithms. So next we will compare the collection, management, and analysis of these versions and show what we did. So comparing Workflow 0 to 1, we added the collection of class definitions as definition provenance and bytecode, and as execution provenance, we also added a tracer for collecting variables as fine-grained provenance. However, the provenance collection of NoWorkflow 1 has some drawbacks. First, the usage of the bytecode is unstable because bytecode of Python may change on patch versions. of Python. Second, the collection of the deployment provenance as that occurs since NoFlow 0 has a problem of causing side effects on the execution, because NoFlow 0 and 1 use the module finder to collect a snapshot of the imported modules in the beginning of the execution, and some scripts may depend on the execution order of the modules, of the imports. Also, the drawbacks in the execution provenance collection are that the way that NoFlow 0 and 1 collects execution provenance is disconnected from the definition provenance. So it shares some names, but it does not guarantee that their names represent the same elements. And the tracer that we implemented for fine-grained collection only receives line events from Python. So it's hard to detect expression dependencies that occur inside a line, and it's hard to support complex data structures. So for NoWorkflow 2, we removed the bytecode collection and we replaced the previous provenance collection based on the passive monitoring strategy to an overriding strategy that overrides the AST during the definition provenance collection. So now we collect all AST nodes, and during this overriding we include provenance collection functions that collect dependencies among evaluations of expressions during the execution, and it also allows us to collect complex data structures and to relate definition provenance to the execution. In addition to these changes, for deployment provenance, we replaced the module finder strategy that collected a snapshot of the modules by a continuous strategy that is based on import hooks. So when the imports are executed, we collect them. Comparing the provenance management, NoWorkflow 1 extended the storage, distribution, reproducibility, and versioning of NoWorkflow 0. So due to the changes on the provenance collection to support the fine-grained provenance, NoWorkflow 1 changed the data model to include tables related to the fine-grained provenance. And it also added the support of exporting the fine-grained provenance as a graph file to distribute the provenance. NoWorkflow 1 also proposed a version model that identifies the intention of the evolution and produce history of versions, and it allows to restore previous trials versions, which can be used for reproducing the experiments. And NoWorkflow 1 also adds a web server that allows the access and visualization of the provenance. For NoWorkflow 2, since we replaced the provenance collection mechanisms, we changed the relational data model, and we also added the ability of exporting provenance as an interoperable format. The prov, but it's actually versioned prov extension. And also NoWorkflow 2 supports a more efficient content database that uses Git for compressing the files. So comparing the analysis, NoWorkflow 1 added a query based on the ORM that uses SQL Alchemy. It is also integrated with ES Workflow to support filtering data flows and running data log queries. And it added a web visualization tool with many types of graphs and support for clustering and filtering them. NoWorkflow 1 also added the support for comparing activation graphs. And comparing NoWorkflow 1 to 2, NoWorkflow 2 removed the integration with ES Workflow, but it added support for a partner matching that is based on the Prolog syntax, and it supports running pattern matching without having to export the provenance to Prolog and without having to install Prolog. And it also supports visualizing the provenance in the interoperable format. Currently, the only tool that supports the version.prov extension is the extensible prov-n that we implemented, but we intend to convert version.prov to prov in the future for proper support in visualizing provenance in other tools. Also, NoWorkflow 2 improved the efficiency of the graph comparison. So in this section, we proposed two approaches that collect, manage, and analyze provenance from scripts. And we got 4 papers in IPOL and 1 VLDB demo. NoWorkflow has been used by other research, and it has been adopted by Core, an infrastructure designed by NIST. In addition to supporting scripts, we also proposed an approach to support provenance in notebooks. So based on the notebook study, we propose best practice for the quality and reproducibility of notebooks. And based on this practice, we propose and evaluate JUlinter, a linter for notebooks that has a Jupyter Lab extension that gives recommendations in real time during the development of the notebooks. And the goal of this recommendation is to improve the quality and reproducibility of the notebooks. We evaluate JUlinter in three phases with 14 participants, and we received mostly positive feedbacks and some suggestions of improvements. In addition to JUlinter, we also proposed the integration of NoWorkflow with Jupyter to collect provenance from notebooks. So we have 2 integrations of NoWorkflow and Jupyter. The first integration is an extension that provides cellmagics for collecting provenance from specific cells in the notebooks and for running queries and analysis. And the extension also provides display methods for visualizing the provenance without having to open the web visualization tool that we proposed before. And the other integration of NoWorkflow was a kernel for Jupyter that collects the provenance of the whole notebook, and this kernel considers cells as activations and the notebook session as a trial. So with this mapping, it's possible to use all other NoWorkflow capabilities for analyzing the provenance, including the notebook extension. And it's also possible to use the provenance for cleaning the notebooks. That is, restoring the cell execution order, like in B here, or producing a valid subset of the notebook based on evaluation that is reproducible. So here in C and D, we are producing a subset that is based on the evaluation of CO in the cell 7, and this is reproducible because it considers all the dependencies that led to the generation of CO. So in this section, we propose best practice for notebooks. We propose a linter tool that uses provenance from proposing recommendations for notebooks, and we propose 2 integrations of workflow to Jupyter. And we published papers in TAP, MSR, and EMSE. So, concluding this presentation, we analyze the state of the practice of the usage of scripts and notebooks, and we run a big reproducibility study to find how reproducible are notebooks and what are the good practices and bad practices. We also run a study on the state of the art of provenance from scripts, and we propose a taxonomy for that. We proposed an approach for collecting, managing, and analyzing provenance from scripts, and we also proposed an approach for collecting, managing, and analyzing provenance from notebooks, and we integrated workflow, that is the approach that collects provenance from scripts to notebooks, and we also proposed JUlinter as an approach that uses the provenance from the simple provenance that already exists in IPython. So there are many opportunities for future work to build on top of the contributions of this thesis. It's possible to create tools based on the insights of the script study. It's possible to run replications on our experiments to try different data sets for the state of the practice studies, or try distant configurations for the reproducibility study of notebooks. It's also possible to improve workflow to support parallelism, collection of black box functions, collection of fine-grained files to consider not only the files as a whole, but consider specific parts of the files. And it's also possible to use the provenance collected by workflow to support debugging, code coverage, improve cleaning, and we also intend to integrate workflow to JUlinter to improve the recommendations of JUlinter. So during the doctorate course, we published 9 papers in the context of the thesis, and we received 2 awards for the MSR paper. And we also published 5 papers in other contexts and received 1 award for a SPES paper. I would like to thank all the collaborators that worked with me and everyone that made this work possible, including my family and the research group meetings. And I would like to thank the committee for reading the thesis and being here today. So thank you.